Hi guys, welcome to GeeklyEDU. This is me, Sona Singh, and today we are going to talk about genes and genomes, what they are and how it is relevant to us in our biology and even daily lives. So let us start right now. So you often see this picture when you are looking about genes and chromosomes in the book. So let us delve exactly what this is and how are genes related to this. So you see sometimes a butterfly shaped structures. Why it's butterfly shaped structure? Why it's two? And what are these two things? So guys, these two things are chromatids, and your chromosomes duplicate themselves as two chromatids, and this is visible only when the DNA is duplicating. That means the cell has undergone or a cell is about to undergo division and it duplicates its DNA and it condenses to form this butterfly shaped structure. This happens only during cell division. So this is when you see chromatids. And what are chromatids? Just condensed form of the DNA. Now, if you further zoom out and look at what these chromatids are made up of, you will see supercoils of stuff. What are these supercoils made up of? Again, they are made up of DNA. You further zoom out in one of the zoomed supercoil, you'll see a coil, and when you zoom out the coil, what you see is a piece of DNA. A piece of DNA in humans and eukaryotes is double-stranded. That means it has two strands which are linked together by bonds. Now let us further zoom out and try to carry out what exactly these are made up of. What are these units? So these units are made up of substances called deoxyribonucleotides, which carry a base, a sugar, and a phosphate, right? But there are two strands, remember that, right? There are two strands. So let me just copy the same three deoxyribonucleotides on the other side as well, so that there are two sequences of deoxyribonucleotides which are linked with each other, but they must be placed in the opposite direction. So that is why I've inverted them, to give you an idea that they are placed in opposite direction. That is why you call your DNA as anti-parallel, right? Because they're placed in opposite directions and there's hydrogen bonds in between them. Okay. Now, what does the opposite direction mean? Just in case you're confused, then sugar, to fill the fifth carbon, we attach a phosphate, right? And so that is called the 5' end, and the other one is a 3' end, because at the third position we attach a hydroxyl OH group, right? Same way, the strand 2, the phosphate is attached downside to the fifth carbon, and to the third carbon is attached the hydroxyl. That is why 5' and 3' end, they run in the opposite directions. Now, what is the difference in these deoxyribonucleotides? So there are 4 varieties of bases you find in your DNA, that is adenine, guanine, thymine, and cytosine, or commonly we call them ATGC, right? So this is how we store all the information. This is how we capture all the information. And the two strands are nothing but sequences of ATGC on both the strands. Now, a segment of the strand that can code for one characteristic is called a gene. So this is where gene comes in.
Okay, now what are the alleles? You often hear the word alleles. So let's say the blood group is coded by the sequence A. C A T T A G G C, just any random sequence. This is not the exact thing. And let us imagine that there's one particular change in a nucleotide. So instead of G, you have a C. But the change is not too much, and this is a variant. This perhaps codes for a different type of enzyme which synthesizes a different type of carbohydrate, and hence you have a variant called allele. So, what are allele? They are gene variants, right? And different alleles can code for the same character but produce different forms. So, you have a different blood groups, right? Now, how does this happens? Now, what happens to these genes? So, genes are not converted, they are read into mRNA called messenger RNAs, and these messenger RNAs are read into proteins. This is a central dogma, and that means a basic rule of biology. And the process was given by Watson and Crick. The first part, that is gene being read and made into mRNA, is called transcription, and mRNA to protein is called translation, because actually the whole thing changes, right? Because there's very basic difference between DNA and RNA. Now, how is this being done? So imagine this double-stranded DNA, but you have to open it. Now, I hope you remember that the DNA, the two strands have directionality, right? Because on the fifth carbon, you attach a phosphate, and on the third, you attach a hydroxyl. Now, RNA polymerase is an enzyme that will synthesize RNA based on DNA, right? So it's polymerase because it adds ribonucleotides. Okay, so ribonucleic acid is a polymer. Now this reads 3 to 5 direction but adds 5 to 3 direction. Okay, now this has its own complications, but I don't want you to fiddle with that right now. We'll leave that for later. Okay, so because it reads 3 to 5 directions, that is called the template strand, and the other one the non-template or coding strand, because the code is actually just that, right? Because of the complementarity rules, it's exactly just that. Now let us try to do that. So this is a template, A T T G A A T C A, just a random sequence, and when it will be coded, you'll have T A A C T T A G T. But remember, we are making an RNA, and RNA doesn't get to have T's. Instead of T's, that means thymine, it needs to have a U, uracil. See, this is one of the fundamental differences between DNA and RNA. It hasn't got T's, it has got U's. Now, this mRNA is read in forms of three. This triplet is called a codon because each codon will stand for a particular amino acid. That is the next step. That's translation. So if you see there are four bases A, T, G, C, and they are read in triplets, so there are 64 possible codes, but only 61 code for an amino acid, whereas the remaining three are stop signals. So there are 61 codons from 64 possible codes, and the three remaining don't code for any amino acid. They're called the stop codons. The three are UAA, UGA, and UAG. Okay, now AUG is a start codon. That means the translation will always kick in from when an AUG is encountered. Okay, and this AUG also codes for the amino acid methionine. Now, there's something that you need to understand right here. AUG is the start codon, and those three were the stop codons. Okay, now this transcription process was happening inside the nucleus. So this needs to come out into the cytoplasm, and this is read by ribosome for translation. It is read by ribosomes in the cytoplasm or attached to the ER, and finally this is made into an amino acid. How? So let's say UUA, it stands for the amino acid leucine. Remember, both UUA and UUG stand for leucine. So there is redundancy in codons, but no ambiguity. No particular codon can code for two amino acids. So tRNA, transfer RNA, they carry different amino acids. So we have around 20 amino acids, and each tRNA, there are 20 type of tRNAs who carry different type of amino acids, and there's a particular code which we call anticodon. So let's say we have to add UUA, right? So the anticodon would read something like AAU, again complementarity. Remember, complementarity works here as well. Okay, so ribosome would be paid a visit by different tRNAs carrying different amino acids. There are 20 such types, but only that tRNA would stick, the one which has the anticodon AAU, and hence leucine would be added. You can add different amino acids based on different sequence of codons. Let's say we have three back-to-back triplets of UUA. You have three different leucine. And once you encounter a stop codon, the translation ends right there and you get a polypeptide. So this is how genomes are transcribed and translated. You make them into polypeptides and then into proteins. This is the central dogma This is how genes work. Okay, so this was the first part. Now, the second part is genomes. What are genomes? So, genomes, if you say, they're a collective set of genes. They're a set of genes and the possible variants for a particular species. Now, the deeper you get, the more complicated the definition becomes. So we have also started to characterize genomes based on time, based on particular tissue, but overall, genomes is just a set of genes of an organism. We want to sequence genomes because we want to understand the functions. We have done this with fungi, animals, different, and most importantly, humans. The Human Genome Project was a big project that was launched by the United States Department of Energy, and a lot of different institutes and universities came together, like the National Institute of Health. Again, the United States came, and they started in around 1990. University of California came in, MIT came in, and a lot of different universities all around the world, from France, from UK, from different countries, came in to contribute to the Human Genome Project. In 2001, the first draft was released, and 2003, this was declared to be complete. This allows us to read the genes and try to understand their functions. And we have deposited all the information at the NCBI, National Center for Biotechnology Information, again hosted by the National Institute of Health. So, this is where we have deposited all the information regarding the genes that we know and we have knowledge of. Now, we also have proteomes, which is a set of proteins in a particular organism, just like genomes. Now, I want you to see how this actually works. So, just Google NCBI. The first thing you'd see up is the National Center for Biotechnology Information. Click on that, and this blue box asks you all databases. Just click on gene and search for anything. For example, you want to talk about hemoglobin gene. Okay, so because NCBI has the genome, human genome, the whole set, you'll see various genes and their description as well. Choose the organism. We want to talk about the human hemoglobin, that is a Homo sapiens. You can see 225 such sequences have been deposited. So these are different variants by different studies. Remember that anyone can submit it, right? So once we chose humans, we could get the human hemoglobins, right? Different chains, right? Now, if you talk about human genome, you can also get the human genome. All of this, right? So this is a whole data set that consists the set of all genes, which is around 20,500 as of our latest understanding. I hope this video helped you to understand all about genes, all about genomes, and how we preserve all this data.
We hope this video was helpful. To see more biology topics covered, click on our channel and don't forget to subscribe.